We'll first start looking at the light reactions. The light reactions are going to take place in the thylakoid membrane. And the thylakoid membrane has pigments in it. The pigments are going to be arranged in clusters called photosystems. The light reaction is going to provide the products of oxygen, right here. It's going to provide reduced NADP. So NADP is a coenzyme that's going to be carrying the hydrogens. And then ATP is going to be synthesized as well. The NADPH is going to be used in the Calvin cycle, and so will the ATP. So we'll look at the Calvin cycle second. So the solar energy, of course, is going to come from the sun, but any light energy can drive photosynthesis. There are two photosystems that are going to be embedded in the thylakoid membrane. So picture these Mentos that are stacked and imagine them to be hollow. The membranes in there contain pigments. The pigments are clustered into what are called photosystems, and in higher plants there are two of them. You're going to find that it's counterintuitive what we're really seeing here. Photosystem 2 acts first, and then photosystem 1 acts second. But the reason they are named like that is they're named in the order that they were discovered, not in the order in which they work. These are pigment complexes, so they're clusters of pigments that are found in the membranes that contain a wide variety of pigments, and the pigments that are in there are dependent upon the organism. So most of organisms will have chlorophyll A and chlorophyll B, and there also is an electron acceptor. This is a molecule. You'll see a video that it describes it a little more detail than your textbook does. And this electron acceptor is going to take the electrons that are lost from these excited chlorophyll molecules. So these photosystems are complexes that actually are acting like antennae. They are gathering as many photons as possible. So if there are carotenoids in there and xanthophylls, chlorophylls, all of them are absorbing different wavelengths of energy. And when they absorb this energy, they begin to vibrate. As they vibrate, then they pass the energy from molecule to molecule. It's like a big dance going on where one starts dancing, the next one starts dancing. It sends that energy to the center of the group. That's the reaction center. And the reaction center chlorophyll molecule, which is a specialized chlorophyll molecule, gets so excited that it loses 2 electrons. These electrons then are going to be passed to that electron acceptor. So photosystem 2 acts first. It absorbs solar energy and energizes electrons. The electrons in the in the reaction center escape to an electron acceptor molecule, and then they are sent through an electron transport chain. The electron transport chain is going to generate ATP by pumping protons. Now, the electrons that are lost from this reaction center have to be replaced. Chlorophyll can't keep losing electrons, so the electrons are going to be lost by splitting water. When water is split, it is split to hydrogen and oxygen, and remember, 1 hydrogen equals 1 electron and 1 proton. So the electrons from hydrogen can go back to replace the electrons lost from the reaction center. What's left is oxygen, and oxygen is going to diffuse through these stomates and is going to leave the plant, and then we can use the oxygen. The electron transport chain that is between these two photosystems is a series of carriers that are capable of passing electrons along. As they pass the electrons along, they are going to synthesize ATP. They pump protons or hydrogen ions across a membrane, the thylakoid membrane, which creates a gradient, and this gradient is used to drive the production of ATP through a process known as chemiosmosis. This chapter doesn't discuss chemiosmosis in a lot of detail. We'll actually see more detail in the next chapter on chemiosmosis. Finally, these electrons, they're pretty tired. They make it to photosystem 1. These electrons are going to be energized again by absorption of solar energy. There are pigments in photosystem 1, and the electrons are going to be captured by another electron acceptor molecule. So in the center of photosystem 1, there is also a specialized chlorophyll molecule that will lose electrons. These electrons are going to be combined with hydrogen and pass to our coenzyme NADP. NADP picks up one electron and one proton to become NADPH, or reduced NADP. The replacement electrons that are lost from the photosystem I reaction center came from the electron transport chain and photosystem II. Okay, so remember that the electrons that replace in photosystem II come from water. The electrons that replace come in photosystem I come from photosystem II. So here we see the entire process, and imagine this entire area here to be the thylakoid membrane. Okay, the thylakoid membrane has two photosystems. Here's photosystem II. Here's photosystem I. Solar energy strikes this photosystem. Molecules become excited. When the molecules become excited, an electron is lost. The electron goes to, you can see we have higher energy here. So electron goes to the electron transport chain. The electron transport chain then synthesizes ATP. Remember that ATP is needed to drive the Calvin cycle. Photosystem II now also is absorbing electrons. It's getting excited. The reaction center releases electrons here. They also go to an electron acceptor, which is NADP. So NADP picks up an electron and a proton to become NADPH. Okay. NADPH, excuse my updates, NADPH is going to be used also in the Calvin cycle. It's going to provide the hydrogens that are added on to carbon dioxide to make the carbohydrate. So when we look at the thylakoid membrane, we first have photosystem II. Between that would be an electron transport system, and then photosystem I. With this arrangement, this promotes the efficient transfer of electrons. We also find another molecule in here called ATP synthase complex. Just by looking at that word, you know what that enzyme does. It synthesizes ATP. Sometimes it's called the lollipop enzyme, and in the video you'll see why. It's a molecule that has a shape very much like a Tootsie Pop. So it's is hollow down the center, and it allows for the flow of hydrogen ions. So what happens is that the thylakoid space becomes a reservoir for hydrogen ions. It has a high concentration of hydrogen ions. Every time water is split to replace the electrons lost in photosystem II, two hydrogens remain in that thylakoid space. The electrons go on to replace the lost electrons, and the oxygen leaves the cell. So now we have two extra hydrogen ions. Energy from electrons transferred between carriers are used to pump more hydrogen ions from the stroma into the thylakoid space. This gives a gradient. Gradients, remember, have a large amount of potential energy, and the energy can be used to make ATP. The hydrogen ions then flow down the concentration gradient. In other words, they're going through this ATP synthase molecule. As they go through this ATP synthase lollipop molecule, the energy release is enough for the production of ATP. This molecule is pretty amazing, and we'll take a look at it in more detail when we get to the next chapter, and that's ATP synthase. Remember NADP, that's the coenzyme. The coenzyme accepts electrons and a proton to become NADPH. So when we look at the products of the light reaction, we see oxygen, NADPH, and ATP. Okay. Oxygen leaves the cell. NADPH and ATP are going to be used by the Calvin cycle.
Membrane. As the protons move through the proton pump, they begin to build up on the Proton pumps are protein complexes that move the protons generated during oxidation reactions across the cell membrane. As the protons move through the proton pump, they begin to build up on the outside of the membrane. The protons accumulate on the outside of the membrane, creating a concentration gradient. The membrane is not permeable to the charged hydrogen ions, and they cannot diffuse back across the membrane. Instead, they must pass through a special channel. The protons move through this special channel, which is the enzyme, ATP synthase. This enzyme uses the energy derived from the movement of these protons to convert ADP into ATP. The movement of protons down a concentration gradient provides the energy for ATP synthase to form ATP. This mechanism of producing ATP is called chemiosmosis.
So can you see why it's called the lollipop enzyme? It looks a lot like a lollipop. And when scientists saw these enzymes in the thylakoid membranes, they didn't quite know what they were, but they found out if they chopped off the heads, that ATP synthesis stopped. So they deduced that they were responsible for making ATP. So it was like biting off the head of the lollipop, and it made stop the production of ATP. Okay, so here we can see the entire thylakoid membrane. Here's the thylakoid membrane. Remember, there's that thylakoid space, and outside of that is the stroma. So we're looking at the chloroplast. Here's the membrane, and the membrane consists of the photosystems. Photosystem II is going to work first, then photosystem I. In between, there is an electron transport chain, and in the electron transport chain, we're going to see pumping of protons. Now, let's not forget what happens here, with the water. Okay, water is going to be split. When water is split, it leaves half a molecule of O2, or just oxygen, and 2 protons. The protons are going to be concentrating in the thylakoid space. So we're going to have a whole lot of hydrogen ions building up here, and you can see then when there are more on one side of the membrane than on the other, they're going to go through the ATP synthase complex. When they go through the ATP synthase complex, they're going to synthesize ATP. ATP is needed in the Calvin cycle. All right, in the meantime now, we've got the electrons that are getting all excited and leaving the reaction center of photosystem II. They are going into the electron transport chain, being passed down. As they do this, ATP is being synthesized, and those electrons now find their way to photosystem I. Photosystem I also loses electrons, and those electrons are going to go to NADP along with hydrogen. That reduces NADP to NADPH, and the NADPH is going to be utilized in the Calvin cycle. Right? So this is a pretty long video, but it describes the process very well.
The photosystems consist of arrays of chlorophyll molecules. Chlorophyll, the green pigment of leaves, absorbs light energy. The absorbed energy excites electrons to a higher energy level. Energized electrons from photosystem I are passed down an electron transport chain and added to NADP+ to form NADPH. Meanwhile, energized electrons from photosystem II are passed through another electron transport chain. Their energy is used to pump hydrogen ions from the stroma into the thylakoid compartment, creating a concentration gradient. Electrons leaving this electron transport chain enter photosystem I, replenishing its lost electrons. Photosystem II replenishes its electrons by splitting water. Hydrogen ions and oxygen are released into the thylakoid compartment. This is where the oxygen gas generated by photosynthesis comes from. The buildup of hydrogen ions inside the thylakoid compartment stores potential energy. This energy is harvested by an enzyme called ATP synthase. As hydrogen ions diffuse through ATP synthase down their concentration gradient, the enzyme uses the energy of the moving ions to make ATP. Next, ATP and NADPH are used in the sugar-making process of the Calvin cycle.
Okay. So the Calvin cycle now is going on in the stroma. So that's the gooey center of the chloroplast. And the necessary components of the Calvin cycle are going to be found there. The whole purpose of the Calvin cycle is to make sugar. It needs ATP and it needs NADPH to do this. It also needs carbon dioxide from the outside. Now remember that the ATP and the NADPH were generated by the light reaction. The end product of the Calvin cycle is glucose, but making glucose requires a couple of steps. The first step is called carbon dioxide fixation. Fixation doesn't mean that carbon dioxide has to be fixed because it's broken, but it means that the carbon dioxide is fixed or stuck to a molecule called ribulose bisphosphate. We sometimes just call it RuBP. The second step is the reduction of carbon dioxide. Remember that reduction is gain. So carbon dioxide gains hydrogens. And then the third step is the regeneration of that first substrate. So RuBP has to be regenerated. So this is a cycle, hence the name, the Calvin cycle. Okay. First step, carbon dioxide from the atmosphere gets in through the stomates. If the plant has stomates, it will open through there, and if it's a simple organism like a cyanobacterium, the carbon dioxide is simply going to diffuse across the cell wall and the cell membrane. The carbon dioxide then diffuses into the chloroplast, and it attaches to RuBP, by an enzyme known as RuBP carboxylase. Its nickname is Rubisco. This 6-carbon molecule, 1 carbon dioxide and 5 carbons in RuBP, that makes a 6-carbon molecule, is very quickly split into 2 3-carbon molecules. Now, carbon dioxide is reduced. The hydrogen and the hydrogen from NADPH is going to give the electrons, and ATP for energy from the light reaction is going to provide the necessary components to reduce carbon dioxide. Okay, remember, reduction is gain. So carbon dioxide is going to gain a hydrogen. The first molecule formed is called glyceraldehyde 3-phosphate. Not the first molecule, the last molecule formed is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate essentially is a half of a glucose molecule. At the very end of the cycle, there has to be a regeneration of the molecule that started it all, and that's ribulose bisphosphate. So one of these glyceraldehyde 3-phosphate molecules can be used to make glucose or other organic molecules. 5 of them have to go back to reform RuBP. Remember, RuBP is a 5-carbon molecule. All of this is going to require energy, and that's the ATP that came from the light reactions. So chloroplasts make their own ATP, but that ATP is going to be used up in the light reactions. That ATP is not available for the plant to do other things. Okay, so here we can see this process. We're looking at it in groups of 3. When you look at the video in a minute, you're going to see they're looking at it in a different light. It might make more sense to you. Here we have 3, there will be 3 molecules of RuBP. RuBP, or ribulose 1,5-bisphosphate, and it's called 1,5-bisphosphate because it has a phosphate on carbon number 1, and it has a phosphate on carbon number 5. That means there are 2, or bi. Bis is another abbreviation for that. So that's ribulose bisphosphate. It's going to fix carbon dioxide. And when it fixes carbon dioxide, you're going to get this intermediate. Okay, the intermediate has 6 carbons. All right. This 6-carbon molecule is very quickly split into 6 3-carbon molecules, and ATP from the light reaction is going to be used in the CO2 reduction. So 6 3-phosphoglycerates are going to be converted into 6 1,3-bisphosphoglycerate molecules. And the good news is you don't have to learn the names of these molecules. Now we've got the NADPH coming in from the light reactions. The light reactions are going to provide the hydrogen in order to reduce this molecule. Okay. What will be made now are 6 molecules of glyceraldehyde 3-phosphate, and one of those 6 is going to leave to go on to make glucose. 5 of them are now going to return to regenerate ribulose bisphosphate. Energy again is going to be needed to do this, and the energy will come from the ATP. So we see that 9 ATP are going to be needed in order to run this cycle, and we're going to need 6 molecules of reduced NADP. Okay. So you can see that this is the Calvin cycle. This is going on in the stroma. And let's take a look at the video. This one is a pretty long video that describes this process.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP, and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just 1 carbon that gets soldered to 1 RUBP chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
Okay, so our word of the day is rubisco, R-U-B-I-S-C-O. Rubisco is most likely the most plentiful protein on the planet. Now, if you think about it, rubisco is the enzyme necessary to fix carbon dioxide to RuBP. So there's a lot of photosynthesis going on, and as you can see from that video, there's a lot of carbon dioxide fixation going on, and rubisco facilitates this, so it is a very, very plentiful molecule. The last thing we want to take a look at in this set of slides is what happens to that glyceraldehyde 3-phosphate molecule. In the video, you saw that it goes to make glucose, but it's not the only thing that glyceraldehyde 3-phosphate can be used for. In fact, plants are so efficient, they can make any molecule they need from glyceraldehyde 3-phosphate. For example, glyceraldehyde 3-phosphate can be used to make amino acids, fatty acids, and glycerol. So plants make oils, as you've learned before, and these oils are going to be made from glyceraldehyde 3-phosphate. Plants also make proteins, and they can do this from glyceraldehyde 3-phosphate. Of course, glyceraldehyde 3-phosphate is going to be used to make glucose, and it also can be used to make sucrose. Remember, sucrose is going to be the transport protein, the transport molecule, it's a carbohydrate in plants. Plants also are going to make starch for storage. So potatoes, for example, have a lot of starch, and the starch is going to be made from glyceraldehyde 3-phosphate. And finally, plants need to make cellulose. Remember, cellulose is a polymer of glucose. So cellulose is going to be made from glucose, which is made from glyceraldehyde 3-phosphate. That ends this set of slides. So there will be a quiz for you to answer on Blackboard, and there's one more set of slides for you to view.